Hi Year 9, so we're back on B4. Last lesson we looked at the uses of glucose in a plant. So we know that plants go through photosynthesis. In that process they make glucose, and we looked at the 5 uses of glucose in the plant. So we looked at respiration, which all living things do, last lesson. We also looked at that glucose being stored as starch. Okay, taking those small glucose molecules, joining them up in a chain to make the longer molecule starch, which the plant is able to store some of that excess glucose as. And maybe when it's dark, they take that starch, break it down to get the glucose back to be using that as a food store at those times. And I've had some really good paragraphs for the conclusion sent to me, so keep those coming, or send them to Sarah if Sarah's teaching you at the moment. Whoever set the work for you, that's who you've got to send those paragraphs to. Okay, so they're really good to read, and I'll be awarding some house points later for those paragraphs. We've got a bit of a quicker lesson today. We're just going to have a quick look at this extension task. Okay, so it's kind of a match-up, reminding us about the iodine solution. So we've got some orange leaves there, we've got an orange and a blue leaf, a kind of zigzag patterned one, and then 2 that are completely blue. So reminder that this means starch is present, and this one over here means no starch is present. And we've got to match up with A, B, C, D and E. And I've given you some help there because soda lime takes away carbon dioxide, so you've got to think about what that might do. Carbon dioxide is needed for photosynthesis, and the hydrogen carbonate provides carbon dioxide. Okay, so let's think about how we might match these up. Spend a bit of time. Okay, if you need extra time, pause now. So plant A, normal conditions. Normal conditions means photosynthesizing, which means storage of starch. So A could be this first one here. Plant covered by black paper strip. I think that's definitely looks like this one here. Then I'm going to skip forward to plant E, which says it's a variegated plant in normal conditions, and that is this one here. And then we've got one where no carbon dioxide is present. So if there's no carbon dioxide, there's not going to be any photosynthesis, therefore there's going to be no starch. So this one here is C. And then plant D, where there is a lot of carbon dioxide being produced. That means photosynthesis can occur, and so that is going to be this leaf just here. Okay, so get those down in your book. Quite good examples to see different aspects of the experiment, what different conditions could affect the results. And if you need extra time, pause now. Okay, so we're back to the same title, I think, as last time, How Do Plants Use Glucose? And we're going to look at those 3 other factors today, so the cellulose, the fats and oils and the amino acids, and then we're going to bring all those ideas together to get the complete picture of the 5 uses of glucose, which I nicknamed SCARF. If you need extra time, pause now. So fats and oils then. Again, the plant has done photosynthesis, it has produced the glucose, and some of it is stored as fats and oils. Think sunflower oil, think olive oil. They come from the plant in those fat stores, okay, the sunflower seeds, the olives. The advantage is that they have loads and loads of energy, more energy stored than in starch, okay, so that's a good thing. But they're difficult to make and difficult to break. A bit like fats in our bodies, they're harder to make and break than just using our carbohydrate stores, okay, which would be our, you know, when we eat carbohydrates, we burn those off. If we eat some pasta or something like that, we burn that off a lot more easily than we do the fat in our body. So again, we've got our glucose molecules. They, rather than forming that long chain like they did with the starch, they've got the long chain, and then they've got these cross branches, okay, and we call fats lipids. So we've got that word just there as well. Okay, so let's get some notes from this page. And if you need extra time, then pause now. Okay, then we've got cellulose production. I said that cellulose was important for cell walls. It strengthens the cell wall, and the cell wall in the plant or any plant is rigid, helps keep its shape, so it's fixed, it's quite strong, and the cellulose helps that. And again, if we look, there's our glucose molecules, and it's just about how they arrange to make that different structure called cellulose. So it kind of looks like two rows of starch that have been joined together with these kind of cross links, okay, and that is our cellulose. And if you need extra time, then please pause now. And then we've got amino acids. So some of that excess glucose is used to make amino acids. Amino acids then join together to make proteins, and those proteins are used for growth and repair in the plant. We can't just say glucose is used for growth in a plant, okay? Glucose is used to make amino acids. Amino acids then join together, and then they are used for growth, okay? So combined with nitrates to make protein. So there's our glucose. And it doesn't look anything like it anymore. There it is, combined with those nitrates to make the amino acids that then build up to make proteins. And then those proteins are used for growth. And if you need any extra time, then pause now. Okay, now what we would normally do in class is I've got this summary table. I'll leave it on here for a minute. It might be something you want to complete. Equally, you don't have to, because we have got all of the notes in our book, so it's kind of like a consolidation activity. You can see we've got SCARF spelt at the top there, uses of glucose from photosynthesis, starch, cellulose, amino acids, respiration, fats and oils. Okay, if you need extra time, then pause now. And then normally I would give out this sheet which has got all of the answers, and there needs to be 3 of these in each of the columns. So if it is something that you want to have all in one place, I know that some people have taken a photograph of this screen, and then you could use that as a cut and stick, or you could copy the information, or if you're happy with the notes that you've got in your book, then that's absolutely fine. You can move on from this. Totally up to you today. If you need extra time on this slide, then pause now. Okay, we're going to look at one exam question, and then we're at the end of the lesson. A student investigated the conditions that plants need for photosynthesis. The leaves of the plant he used had green and white parts, so it's a variegated leaf. Diagram 1 shows how part of one leaf was covered in black, opaque card. The plant was placed in a warm, sunny area and was watered well. 8 hours later, the leaf was removed from the plant and was tested for starch. The results of the test are shown in diagram 2. The shaded parts show where starch was present. Okay, so you can see that it's even labelled starch present, original position of the card, and we've got a part labelled A and a part labelled B. The outer part of the leaf is white, green in the middle. Here was the black card. Starch here, we've got A and we've got B. Let's have a look at the question. Name the 2 independent variables in this investigation. They're actually looking at quite a lot of stuff here. There's more than one thing going on at a time here. So there's 2 independent variables. What 2 things did they change in this investigation? Have a think. Okay, if you need extra time, pause now. Why was no starch found in the part labelled A? Okay, why was no starch found in the part labelled A? If you need extra time, pause now. was no starch found in the part labelled B? Why was no starch found in the part labelled B? Need extra time, pause now. Okay, so green pen time. So the first question was, what were the two independent variables? We had light intensity, because we had the reference to the fact that it was covered or uncovered, so we can't just say part of it was covered, part of it was covered. Part of it had no light and part of it had light, and part of it where it was variegated leaf, part of it had chlorophyll and part of it had no chlorophyll, or you could say chloroplasts and no chloroplasts. Okay, and why was there no starch found in A? That was the bit under the card. It's because it did not receive any light, or it's in the dark, so therefore no photosynthesis could occur. And the bit on the outer edge labelled B, just there, that's the variegated part, the white part of the leaf. That was because there's no chlorophyll, no chloroplast present, so therefore no photosynthesis can occur. If you remember, we've talked previously in the topic, we said that the light and the chlorophyll are like the ovens. If we don't have the ovens, we can't bake the cake. Okay, so without light, no photosynthesis. Without chlorophyll, no photosynthesis. If you need any extra time to do your green pen, then please pause now. Okay, that takes us to the end of our lesson today. So we've done lots and lots of work now on photosynthesis, and we'll be moving on in the next part of the topic next week. Okay, we've gone down to 3 lessons this week with the new timetable, so this finishes us off nicely for the day. Bit of a shorter lesson than last time. Okay, hope you found that useful. Thank you.